Une, deux, une, deux. Une, deux, une, deux, une, deux.
Ok. Donc si je veux le mettre lui en full, je dois faire ça.
Oh Vanessa, ça suffit. Vous savez, je préfère.
C'est un beau prénom. Mais évidemment, avec le mien, peut-être que c'est encore mieux.
Il paraît que... Il y a une boucle.
Où ça?
Je sais pas, ici.
Non, t'as une boucle quand tu mets la caméra. Il n'y a pas besoin de mettre la caméra quand... Il n'y a pas besoin de mettre la caméra ici quand on est de l'autre côté. Disons que... Il faut laisser la caméra pour l'instant, mais quand on mettra le gars en présentation, il faut couper la caméra qui est ici, quoi. Ce qui est pas... La personne à distance. Je ne suis pas...
Je repasse la parole à Stéphane.
Une, deux, oh, 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 oh. Un, deux.
1, 2, 1, 2. Micro 2, micro 2, oh, 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 oh. Micro 2, micro 2, micro 2.
Il va pas fort, mais il fonctionne. Donc je te le mets sur le mute pour l'instant.
Une, deux. Madame, Messieurs, bonjour.
Good morning, I am here. Ecco, ok.
Donc toi, tu peux lui parler. Oui.
Bonjour. Bonjour, comment allez-vous? Vous m'entendez?
Buongiorno professore.
Bonjour, bonjour.
Bonjour.
Vanessa, je regrette.
Abbiamo un problema. Yes, Sébastien.
Il y a... Voilà.
Voilà.
Professore?
Sì,
Ah, adesso è
buongiorno,
bello, ci
buongiorno.
sentiamo.
Certo. Mi dispiace tantissimo, purtroppo, questo incidente del Covid, ma...
E mi dispiace allontanarmi.
No, c'è un eco grandissimo. Non la sento. Il suo microfono è chiuso, Vanessa. Volevo provare a vedere se potevo condividere lo schermo per vedere se va tutto bene. Però non la sento. Ecco, adesso no, il suo microfono è chiuso sempre. I cannot talk. Yes, I know. Your microphone is closed. You have to activate.
Professore Genazzani, mi sente?
Sì? Sì, adesso
Sì.
la sento.
Adesso
Perfetto.
sì, va bene, grazie.
Volevo provare un attimo a condividere il mio schermo per vedere se va tutto bene. Can I try? Jean Michel, bonjour.
Buongiorno professore.
Buongiorno.
Vuole provare a condividere lo schermo?
Proviamo un attimo. Perfetto, proviamo a vedere questa qua.
Oui, il veut essayer de partager son écran pour voir si ça marche.
Voilà.
Funziona, professore, sì, funziona.
Benissimo, grazie mille. Ok, perfetto.
Tutto bene.
Grazie,
Tutto...
benissimo.
Sì, sì, funziona bene. Ci vediamo più tardi.
Ci vediamo più tardi, grazie mille. Ecco, vi seguo tutti.
You tell me when I start. We go. I do? Okay. Messieurs les secrétaires perpétuels honoraires, mesdames et messieurs les membres de l'Académie, mesdames et messieurs, it's a great pleasure today to open the symposium of the Royal Academy of Medicine of Belgium, dedicated to the scientific, medical, and academic activity of Professor Jean Michel Foidart, who is our honorary permanent secretary that just finished his term, and a number of extraordinary and special scientists and physicians came from far away, as well as from Belgium, to honor the activity of Professor Jean Michel Foidart. I will not be very long, but I cannot stop myself in mentioning that Jean Michel Foidart is a very high-level scientist in biochemistry and cell biology, which are basic areas of research in medicine. He has been contributed to many discoveries, for example, laminin, which is a textbook notion, and other things linked to membrane metalloproteases. So he is an accomplished cell and molecular and biochemical researcher that is also a very high-level clinician, and he was able to take fundamental observations that are done in a biochemistry setting and transform them into concepts that are now practically used by patients. So usually we all pay this tribute to the bench-to-bed transition, but in this case is real. It happened, and it happened for patients not only in our clinics, not only in a small clinical trial, but worldwide. So it has been a great honor for me to serve in the Bureau when Jean-Michel Foidart was permanent secretary, and with that I congratulate him for his activity, and I pass the word to Professor Georges Casimir, our current permanent secretary.
Dear colleagues, ladies and gentlemen, Mr. Honorary Permanent Secretary, dear Professor Foidart, allow me the familiarity of the first name, dear Jean-Michel. For 5 years, you have been the 14th perpetual secretary of this illustrious company since 1841. But times have changed. The number of annual advices of the company asked by the government has been increased by 4 in recent years. The pandemic has placed on the shoulder of the Academy major responsibilities that it has had to face quickly. The French community of Belgium, confronted with the planning of the medical offer, has entrusted our company with it. All these responsibilities and many more, such as maintaining the scientific level of our session, you have taken up with brilliance, intelligence, and firmness as it should be. it seems essential to us to thank you for having thus maintained, such as the Orders of Lords in England, the Sword of England at arm's length during the speech from the throne, the quality of the Royal Academy of Medicine of Belgium. And for this, of all solemnly give you the Medal of the Academy bearing the face of its founder, King Leopold I, and on the reverse your name as a badge of thanks from the company for rendered services. And the President will give you now the Medal of the Academy. We saw that honoring you went through the organization of a symposium on the scientific work that has fascinated you throughout your life and also resulted in the creation of one of the fastest growing Belgian pharmaceutical companies today. Mitra notably produces synthetic steroids, but not only. You will have always been, with much happiness and efficiency, a researcher, a teacher, a head of department, a perpetual secretary, officiating in all these clauses with ease and rigor. Your career has been a tremendous success, and the Academy can take pride in you for having been led by a remarkable organizer. But it seems to me judicious to let you now introduce this session with all the skills that include yours.
Tu peux me donner les diapositives? Monsieur le Président, Monsieur le Secrétaire perpétuel, Monsieur le Premier Vice-président, Mesdames et Messieurs les académiciens, dear ladies and gentlemen, I must say that I am extremely happy and honored to have so many nice people and friends today in this audience. I would want to really thank the Academy of Medicine for having organized this session, but you know, I am not very important. What is important is molecular biology, clinical sciences, and the future of our teaching and developing new activities in the field of human health. I would want to very briefly summarize the fact that my scientific career was really influenced by two very important scientists. One was George Martin on the right-hand side from the National Institute of Dental Research. I spent 4 years doing dental research. And on the left-hand side is Stephen Katz. Steve Katz was at that time a very young chief of dermatology at the NIH in Bethesda, Maryland. And both of them were really supervising my research. This research started really with the Danish scientist called Engelbert Holmes. He captured these wild mice who had a small tumors, and he described that this tumor was really composed of a sarcoma. George Martin had really the impression that in order to identify new molecules and new important proteins, you have not to look at normal tissues, but at malignant tissues. He said, okay, when you have a tumor, you have a clonal expansion of malignant cells who produce always the same proteins, and they're all alike, and therefore you have a much higher chance to isolate new proteins. Let's make an example. In immunology, There were at that time in the 1970s, many scientists who had tried to isolate the immunoglobulin and to understand what it was about. And no one had succeeded from immunoglobulin obtained from the plasma. It's only those people who were looking at myeloma or Waldenström's disease with the monoclonal expansion on one type of IgM or IgG who were successful to isolate and understand what is an antibody. So we started looking at the proteins contained in that little mouse, and it turned that it was, I was really the first to develop antibodies to that and to realize that it was really locating the proteins from the tumors in the basement membrane. Basement membrane were unknown, and the composition of the basement membrane were unknown at that time. And I had the fortune to be associated with the discovery of laminin, and the very first paper on laminin was cited more than 3,000 times. And when I returned to Belgium, I started doing research on extracellular matrix and membrane metalloproteases. And my very first PhD student, who is now a member of the Academy of Medicine, I just called her today, is Agnès Noël. She deserves really to be nominated. I called her just a few minutes ago my first professional wife. She is really an outstanding scientist. She was very supportive of all the studies we were developing together. I was first her leader, and then she became my leader. She is really outstanding, and in addition, she is a very nice and sweet person. Then subsequently, many other members of the Academy nowadays are visited the lab, performed their PhD studies or their post-PhD training, like Didier Cataldo, Vincent Castronovo, Rakik, who is an ophthalmologist. He is a basic scientist, and he is a pneumologist. So we had the opportunity with Agnès to develop really basic sciences in the Faculty of Medicine at the University of Liège. But in the field of obstetrics, and I was teaching cell biology and molecular biology for more than 15 years, something like 17 years. Then I was offered to go back to the clinics and to develop a large department of obstetrics and gynecology. And again, there I benefited of the support and the help of a wonderful endocrinologist called Ulysse Gaspard, who is in the audience today. And my second professional wife, who is a clinician, is also a member of the Academy. It's Professor Nizol. Michel Nizol is a fantastic scientist, worldwide known for her expertise in the field of endometriosis. And all the other people, the little guy that you see at the bottom will speak here at the Academy, Kridelka, in just next Saturday. And of course, there are many other people who are candidate cited, but who were really members of the research team. And in the field of Estetrol, there are many people who are now working either together or in Mitra, like Maud Jost on the left-hand side, Céline Gérard, you've seen also Anne Gallet, Christelle Pequeux. The three first are now members of the Mitra team. The Belgian people know very well Blancpain. Cédric Blancpain is really at the cutting edge of science in painting mice, and by using very sophisticated method, he can paint in vivo the luminal cells of the breast, the basal cells of the breast, normal or malignant, and any cell type that you want, he can specifically paint them with at least 12 different colors. So we are actively working, and we have dedicated €1 million to combine research, coordinated research with Christelle Pequeux, Agnès Noël, Cédric Blancpain, and his co-workers, Alexandra van Kemelen. I must also cite a collaborative research that we are doing with another member of the Academy who is also in the audience, Philippe Lefebvre. Estetrol is a neuroprotective agent. It's a neurotrophic agent. We have shown that in just young mice in postpuberty, and just after a few days after birth, those mice, when they are treated with estetrol, are protected against brain damage upon hypoxia. When you place the mice in a hypoxic chamber, those little pups will die, and they have huge brain damage. If you give them estetrol, it doesn't do anything. And they go well, and they feed, and they can find their mother, their way, they are intelligent, and so on. It forced us to say maybe this estetrol could be a neurotrophic agent also for people who become progressively deaf. And Philippe is the chairman of ear, nose, and throat at the University of Liège, and we are actively working on estetrol for the people who are losing progressively their capacity to hear. So, but today we are here to know and develop the session on estetrol. Estetrol was really developed in conjunction with Mitra. At the time we created Mitra in 1999 with François Fornery. It was not the Belgian habit to do it. When I went to the rector of the university to explain that I wanted to create a spin-off of the university, he said, I'm not in favor of that. You should stay away from the business people. They are not good. We don't want to have such dialogue. Says, just stay away from that. I decided to do it anyway. We just rented a small apartment in that building. It was very small. We had just one bedroom who was used by the secretary. And we first developed an intrauterine system that would release levonorgestrel. I had €1 million, but you need to get it up to the clinic, you need 150 million. And we had no money, no cash to do it. So we were stuck. I didn't know what to do with it. And suddenly, Warren Buffett, who had decided to create a charity foundation called Medicine 360, said, okay, this system is really good. That's the best. I'm going to provide you the finances. I will open all US academies, all US university and research centers, and we will do it together with the benefits from the US women and from the US market. We will collect a lot of money that will be used to distribute freely this intrauterine system to the developing countries and to the poor women in the United States. So it was a fantastic part of my professional life, because when you go to FDA and you say, I work for the poor people, they say, well, you work for the Medicine 360 and Warren Buffett, that's great. But the greatest aspect was that I did discover this guy. He is in the audience, and really he has become a great friend. Mitch Kreinen, Professor Mitchell Kreinen, thank you for being with us today. That was the start of a wonderful friendships and collaboration. And it started with the intrauterine system, but subsequently to the development of the contraceptive and of estetrol. And as you know, estetrol is produced by the human fetal liver. It's human specific. It's not produced by other mammalians. Throughout evolution, E4 was absent. And suddenly, when the human separated from the other primates, E4 did developed by the development of specific enzymes that are present in the fetal liver and that will become silent after birth. It crosses the placenta. Okay, Jean-Francois Arnault will tell you that, in fact, you find a little bit of estetrol in the large monkeys, but it's only present in 1% during the last 6 weeks of pregnancy, while in the human, it's produced, the human fetus produces about 100% more, and it starts from the 9th week's gestation. The fetal plasma are about 12 to 20-fold higher. It has a very prolonged half-life, And now, of course, we produce it from plants by synthetic pathways. In fact, the estrogens that we are developing, it's really a highly demanded molecule because the currently used estrogens are very old molecules. In 1929, 1930, 1936, and 1941 and 43, where the time you see that within a frame of 15 years, all the estrogens were developed and used in the clinic, and there have been no significant improvement for almost 80 years. What makes Estetrol really unique? We are supposed to see Arnal pictures, but Jean-Francois is there, so you can see not only the pictures, but the reality. Jean-Francois Arnault is a fantastic cell physiologist, a molecular biologist. He works at the University of Toulouse. He has an INSERM unit. We became friends. We met by accident in 2009. We became very much friends, and since we are working on Estetrol, and he was really the person who did decipher the mode of action. And as you know, Estetrol is really activating the nuclear estrogen receptor alpha, but is blocking the estrogen receptor alpha at the cell surface. He will explain that to you. And finally, we did develop since 2015. I have retired from the university. I'm closely interacting with the Mitra people. I'm still a member of the board, and there are many, many other people who contributed to work. I must say that I'm also very happy of my last professional wife, who was my last PhD student. I had about 98 PhDs during my career, and Dr. Moonen, or it's there, Marie Moonen, is there with us, and I'm very pleased to say that almost 2 more and I would have had 100 PhDs. But you are the last. Thank you for coming. And finally, I want to say that nothing is really possible if you don't have also a personal life and a happiness with your wife. It's my best journey, and I used to say it's my best action in my life is to have been selected by my wife. And those are my 4 children who were very supportive. Thank you very much to all of you for being today and for celebrating the journey to Estetrol. Thank you.
Thank you very much for this wonderful lecture, and I want to say that all the students that may watch us, the key paper on laminin was published in Journal of Biological Chemistry, right? And it is one of the highest cited papers in cell biology. There are examples of Nobel Prize winners that published in JBC, like Brown and Goldstein. So I think that if the science is really, really good, that's what's important, not running after impact factors and keeping the data for years in the drawers. And I think that's really, really important. Now, we have a wonderful session ahead of us, an exciting session with a lot of science. And before that, I want to ask Madame Vanessa Costanzo to open the electronic and digital and media part of the session.
Thank you, Mr. President. So just a quick reminder for everyone of the technical aspects. As always, the Academy will try to make this session as interactive as possible. And speakers will have 10 minutes to answer your questions after the conference. For the members of the ARMB following us on Zoom, you know that you have to write your questions in the Q&A section so that you can either read them or we can either read them or give you the floor to ask them live. And as you see now on your screen, don't forget to allow the technical manager to connect you as a speaker when you see this message appear on your computer. If you are watching us on YouTube, you can send your question to the speakers in the chat box. Please mention your first, last name and affiliation. Today's session will benefit of an accredit from INAMI, and I will let you know when you can send your emails. And with with this, it is more than time to explore this fascinating program elaborated by Professor Foidart. As he said, the journey will start with the molecular biology of estrogen receptors and Estetrol. Mr. President, it is your honor to introduce the first speaker.
Thank you very much. The first speaker is Professor Jean-Francois Arnault. He is an MD PhD. He's a professor of medical physiology at the Paul Sabatier University in Toulouse. He is also a senior scientist at the Institut des maladies métaboliques et cardiovasculaires of INSERM. He is also a senior member of the University Institute of France and also, obviously, a foreign member of our company. He has been president of the French Society of Atherosclerosis, and he has a team that is very well known worldwide for the last 25 years in the basic knowledge of physiology and medicine of estrogens and their receptors, and made major contributions in the understanding of these treatments and also the new classes of SERMs, the selective regulators of estrogen receptors and progesterone receptors. So, Professor Arnault, thank you very much for being with us today.
Thank you, Mr. President. Dear colleague, it's really... Okay. Thank you. Yes, so it's really a pleasure and an honor today to participate to this celebration in the honor of Jean Michel Foidart. So I will try to select what can explain, in fact, the mechanism of action of estrogen at the level of the molecular biology. So you're here. The pointer is not appearing. It's okay. Okay, thank you. Thank you. So here is the structure of the main estrogens, and the estradiol has here a planar structure, hydrophobic structure with two hydroxyl group, and estriol and estetrol has 3 and 4 hydroxyl group in their structure. And what you know is that estrogen receptor is a nuclear receptor and transcription factor. And this is the classic scheme with the estrogen receptor here, its translocation to the nucleus, and most of the action of estrogen receptor until recently were known as a transcription factor here, after binding the DNA here, and eliciting gene expression. The estrogen has pleiotropic effects in women that can be modelized in mouse. And the beneficial one in women, for instance, the prevention of coronary events before 60 years, prevention of diabetes type 2 or osteoporosis, can be mimicked here in mouse models, as well as the proliferative actions of estrogens at the level of the breast or of the endometrium that can be deleterious as they promote, they can promote cancer, can be opposed in the endometrium with progestin. And finally, the action of estrogen at the level of the liver through an induction of coagulation factor can promote, can increase the risk of venous thrombosis. And thanks to this model, we have tried to understand the mechanism of action of estrogen in vivo, mainly in mouse models, and we focused on vascular aspects, on arterial aspects that are mainly protective. And with Francis Bayard, 20 years ago, we began to modelize this in mouse, and we found a huge protective effect of estrogen on atheroma, a prevention of the neointimal hyperplasia, it is to say a prevention of the restenosis of an artery after injury. And surprisingly, we also found a very important role of estradiol in allowing flow-mediating remodeling, it is to say an increase in the size of the arterioles in response to an increase of flow to match with the tissue demand. We collaborated, we began a fruitful collaboration with Pierre Chambon in Strasbourg. And at that time, one question was, because there are two estrogen receptors, ER alpha and ER beta, what is the most important one? Because there was a controversy at that time due to a leakage, in fact, of the first mouse model of inactivation of ER alpha that was done by Ken Korak. And because of a leakage of this mouse, the inactivation was incomplete, and so there was still a chimera of receptor that was still produced and that accounted for some actions of residual action of estrogens. And thanks to a second generation model generated in Strasbourg, we were able to demonstrate, there's the picture of Pr Jean here that you cannot see, sorry. And thanks to this model, we were able to demonstrate that ER alpha, in fact, mediates most of the action of estrogen in vivo. So we focused on ER alpha, and we tried to understand how it works. In fact, ER alpha here is structured with a DNA binding domain, which binds to the DNA, ligand binding domain, which bind the estrogen here, and with the conformation that the binding induces to hold the molecule. And these changes in conformation will activate here AF1 and AF2, that are the two main activation functions of the molecule. Here you have the dimer, which once activated after binding estradiol, will recruit here cofactors, and in fine will recruit the RNA polymerase and will induce numerous gene expression and in fine the biological action of estrogens. So we continue this work with Pr Jean, and we inactivated respectively and selectively AF1 and AF2, the two activation function, and we found that whereas AF1 has just a sensitizing role to estrogen, AF2 is absolutely required, and if you kill, by mutating several amino acids here, if you kill the AF2 function, you kill most of the action, all the action of estrogens, and the phenotype of this mouse is quite similar to the full knockout of ER alpha. So at this step, we found that for all these action, and as well as numerous other action at the level of bone, at the level of the uterus, you have no longer any action of estradiol. And in fact, one key was given by this fourth model of pathophysiological artery with carotid, with a model of carotid artery injury. It consists in destroying both endothelial and smooth muscle on a precise length of 4 millimeters, and then you treat the mouse with estradiol here. This accelerates the reendothelialization compared to an animal that is not given estradiol after castration. And you can quantify this process. You can see here acceleration of reendothelialization by estradiol in wild-type mice. This effect is completely abolished, as I mentioned before, in ER alpha deficient mice, in mice without ER alpha. The normal action of estradiol in mice with ER beta. And very surprisingly, in mice where we have killed the AF2 function, the nuclear action of estrogen, we still have a response to estradiol. And this is the beginning of one new aspect of estrogen. Because if you kill here the action of the receptor in the nucleus, it remains, in fact, subpopulation of estrogen receptor outside the nucleus. And this was studied 10 years ago by John Katzenbogen, thanks to a very original molecule that is estrogen dendrimer conjugate. It is a macromolecule with numerous estrogen bound to this macromolecule, and this macromolecule can enter the cell and stimulate the membrane receptor, but is too large to enter the nucleus. And thanks to this molecule, he was able to demonstrate that activation of this membrane receptor here is extranuclear receptor is able to accelerate reendothelialization. Then we raise the question, is membrane estrogen receptor necessary for this effect? And to answer this question, we mutated, in fact, here in the estrogen receptor one amino acid that is responsible for palmitoylation. Palmitoylation is a lipidation, a modification of the estrogen receptor that allows it to be targeted to the plasma membrane and to remain so outside the nucleus. And we found that after killing this membrane receptor, we abrogated the action of estradiol on reendothelialization, whereas the uterine growth is completely normal. So here, estrogen receptor at the membrane is dispensable for uterine action. And conversely, you have here again the mouse AF20, where the nuclear action are killed, and you still have an acceleration of reendothelialization thanks to the remaining receptor at the membrane here, but the uterine remain atrophied. So let's now move to the Estetrol, and how can we explain the action of Estetrol thanks to this molecular background? So Jean Michel Foidart already mentioned this aspect of estradiol, and one very important point is that Estetrol, that is very abundant here in the human plasma fetus, its role is completely unknown from a physiological point of view. So we try to understand what is the effect of estrogen estradiol and estetrol after binding to estrogen receptor through crystal structure binding approaches or through binding approaches. And when you study the conformation of the receptor after binding of these two estrogens, you cannot, in fact, really find differences. So when you isolate the nuclear estrogen receptor, there is no real difference. Similarly, when you study the effect on uterus, we previously found, and that transgenic mouse and pharmacological model demonstrated that the uterine growth, the endometrial proliferation, relies entirely on the nuclear ER-alpha action. And accordingly, Estetrol here has the same effect on gene expression at the level of the uterus than estradiol. So again, the effect of estradiol and Estetrol on uterus are very similar. And one major difference was given by the fourth model of vascular injury. It is to say the model allowing to study reendothelialization. And again, you see that estradiol is able, as estriol, is able to accelerate reendothelialization through the stimulation of the membrane receptor, but not Estetrol. And if you add simultaneously estradiol and Estetrol, you even block the reendothelialization by Estetrol. And thus, Estetrol differs profoundly from estradiol because it does not activate the membrane receptor, but it is also able to block the action of estradiol on the membrane receptor. And so, first, to summarize these different aspects, we can say that, and I hope I have convinced you, that estradiol is able to activate both membrane and nuclear receptor, whereas Estetrol acts only on nuclear receptor and is not able to activate the membrane receptor, but is even able to block the estrogen action on the membrane receptor. And the counterpart of that in vivo is that some tissue rely mainly on nuclear action, for instance, the uterus. Some endothelial action as reendothelialization, endothelial healing, rely mainly on membrane receptor, and other tissue, as the liver, is a mixed tissue that depends both on nuclear and on membrane actions. And this is the first level of explanation of what we will see later. It is to say the little effect of estetrol at the level of the liver. And how can a molecule bind differently from the same product of a gene? It's probably here the palmitoylation and the numerous molecular interaction of this receptor that allow this entity to be recognized differently by estetrol and estradiol. Is it so simple? Not really, in fact. And to conclude, these are very unpublished data here, where you can see, thanks to single cell gene expression here, single cell RNA-seq, that allow you to characterize, in fact, gene expression in the cells of the aortic vessel. And you can see that you can characterize, so this different population, endothelial cells, smooth muscle cells, fibroblasts, and macrophages. And you can see here that estradiol and estetrol induces gene expression changes very differently here in this subpopulation. A lot of changes here in the macrophage in response to estradiol, for instance, and a lot of changes in smooth muscle cell or fibroblasts in response to estetrol. And so now we have to investigate further why these gene expression changes are so different. And the epilogue of this that is that in a different model of injury, it is to say now in a model where you kill only the endothelial cell and not the smooth muscle cells here, which gene expression is differently changed by estetrol. In that case, estradiol and estetrol both accelerate reendothelialization, and this acceleration of reendothelialization in that case by estetrol is not the consequence of an effect on the membrane receptor, but a direct effect on the at the level of the ER alpha nuclear receptor in smooth muscle cells, because if you kill selectively the ER alpha in the smooth muscle cell, you abrogate the action of estetrol. So, to conclude, estradiol elicits both nuclear and membrane action, whereas estetrol only activates nuclear actions and not the membrane action. And depending on the tissue and cells, you can have similar action at the level of the uterus, a lesser action, lesser hepatic effect by estetrol, and in arteries, it's as most of the action on nuclear, you have the similar protection by estetrol than by estradiol, and with some mechanism that remains to be elucidated. So, estetrol appears as a singular natural estrogen whose physiological action remains to be discovered, but now the medical use will be detailed. And I would like to thank all the people, in fact, that contributed to this work. And in particular, I will now switch to French for 1 or 2 minutes. Je voudrais terminer par, si on enregistre un peu plus personnel, pour Jean-Michel Foidart. Donc, un des bonheurs que m'a réservé ce parcours de médecin-chercheur a été la rencontre de personnes remarquables, inspirantes, avec qui j'ai eu le privilège de travailler, et vous en avez vu un certain nombre ici. Ainsi, il y a 13 ans, j'ai rencontré Jean-Michel Foidart, grâce à un congrès organisé par Andrea Genazzani à Pise. J'ai aussitôt été attiré par sa prodigieuse connaissance de la gynécologie obstétrique, mais aussi par son extraordinaire curiosité concernant les aspects moléculaires. Et ces modèles et ces outils que nous avons développés à Toulouse nous ont permis de répondre, enfin je l'espère, au moins en partie, à un certain nombre de mécanismes de l'estétrol et ainsi à répondre à certaines de ces attentes. Mais bien des mystères demeurent, au premier rang desquels le rôle physiologique de l'estétrol pendant la grossesse. Il y a donc encore du travail pour nos successeurs. Durant toutes ces années, j'ai pu apprécier ses multiples qualités, à la fois remarquable médecin, très grand chercheur, fantastique organisateur et véritable homme orchestre dont témoignent, entre autres, ses actions dans cette académie. Cher Jean-Michel, Je tenais donc à te remercier chaleureusement dans notre langue commune pour tout ce que tu nous as apporté, tant sur le plan professionnel que personnel.
Thank you very much for this amazing lecture. We learned a lot, and we all have a lot of questions. So the way we will manage this is first we will ask questions from the audience here that is physically present, and then Madame Costanzo will manage the questions from Academy members that are on Zoom and public. So questions from the audience. Yes, Patrice Cagny.
Thank you for this very nice lecture, very interesting. I have a very basic question. I'm not familiar with this, this topic. I was wondering if the palmitoylation of the receptor at the membrane space, if we know how it is regulated first, and if some nutritional conditions can change the activity and finally can contribute to explain some of the relationship between obesity, type 2 diabetes, and this ER alpha at the membrane site. Thank you.
Thank you for this question. It's obviously a very good question, a very important question. In fact, the first point is that the membrane receptor is not abundant at all. It's very difficult to isolate this receptor, and even it's very difficult to see this receptor. We have tried by many ways, by adding a tag on this receptor through antibodies, and we have a lot of trouble just to see this receptor, and so it's not easy to characterize its abundance. So we have indirect evidence of this receptor, many evidences, but the direct visualization is still very, very difficult, I would say, not reliable so far. And so the palmitoylation is a reversible modification, and this palmitoylation, the regulation of this palmitoylation is not really, palmitoylation concerns many other molecules, so we don't have the possibility to really investigate the action of changing the abundance of palmitate, for instance, or the enzyme that lead to this post-translational modification. So it's an open question.
Thank you very much, Professor Casimir.
Yes, thank you for this fascinating presentation. My question is, do you have the same side effect with Estetrol given in male animals than other estrogen in male animals?
So the male issue is an important one, because you know that, in fact, males have a receptor almost as abundant as in females. And we don't have studied extensively the action on males. We have studied mainly females. But as for other estrogens, it seems that you could have most of the action in males than in female. Sometimes you're obliged to double the dose, because males are a bit less sensitive, or probably epigenetic modification. And so we can expect, or to have, I would say, rather similar action.
Thank you. When we teach in cell biology this chapter, we talk about some chaperones that bind these receptors. Do you have any evidence whether the chaperones that bind the membrane one versus the cytosolic moving to the nucleus one are different?
Again, it's like for the palmitoylation. It's very difficult to isolate these molecular complexes, because they are very labile, and so it's all this interaction appears very transient. So it has been rather easy and more easy now to study what's happened at the level of the nucleus, and we have many tools for that. Outside the nucleus, it's it's much more difficult, because it's probably submitted to phosphorylations, And these effects are very transient. So it's very difficult to have a picture with time.
But the model eventually is that even this membrane receptor with other partners ends up also regulating genes, right? Yes. So on the chromatin, it should have a different complex.
So there have been many studies, mainly in vitro, trying to see what are the interaction between membrane and nuclear action. And so obviously there is some impact of the membrane action to the nuclear action. One additional possibility that is nicely worked by a colleague in Rennes, in France, is that this action could occur at the level of the RNA. And so the translation could also be influenced by estrogen. So I would like to conclude with the fact that the estrogen receptor appeared very early in evolution. It was the first, it's the father, in fact, of all the steroid receptor. And it's probably the reason why it's so nicely integrated in our physiology.
Thank you very much. Well, it's time also for the outside world to have access.
Yes, but the outside world seems to be very quiet this morning. So I guess maybe we will have questions afterwards. But if we have no more question, no further question here in the room, in the audience, maybe we can switch to the next panelist, and we will take a step further and start talking about reproduction. And to do so, we will have to virtually travel to Italy.
So it's a great pleasure to introduce Professor Andrea Riccardo Genazzani. He is born in Florence. He did his medical studies in Siena University, then became a gynecologist and an obstetrics and specialist. Then he worked in endocrinology and metabolism. In 1972, he received a PhD in clinical biochemistry at University of Lausanne in Switzerland. For all his research, he was awarded a number of prizes, and I am particularly touched by the fact that 2 Romanian universities gave him Doctor Honoris Causa, among which the University of Bucharest, which is the best university in Romania, also University of Athens in Greece. He has been president and officer of many European and international societies. He has more than 830 papers and 45 books. And with that, I thank him really very much for being with us today, even virtually, as he unfortunately had COVID and had to be not coming here today. Thank you.
Professore, non lo sentiamo. Professore?
Yes, the microphone is now ongoing. I'm sorry.
Yes.
Yes, it is happened. I'm sorry. Yes, I am really, as you can imagine, I am really sorry not to be with you, mostly because I have lost a fantastic opportunity to be close to Jean Michel Foidart, to join back at the Royal Academy of Medicine that honored me with my membership, and also to have the pleasure to share my time with you, and mostly to share my affection with Jean-Michel. Today I will speak about you on a topic that I am working on in these times, is estrogens and female brain. We are always, as a man, we are always fascinated what are estrogens effect on the brain, on female brain, and what they should be the difference. And then I will, I will start by that consideration. This is something that we were not so aware in my younger time, that the fact the brain intracerebral content of steroid depend from the output coming from peripheral endocrine glands, but also depend from a local synthesis. Either central nervous system as well peripheral nervous system are both able to metabolize cholesterol to pregnenolone, and from pregnenolone, according to the expression of selective enzyme important for the steroid metabolism to produce selective steroids inside to the brain. The brain is a steroid-producing organ, as is also the adrenal gland or the ovary, but it don't need any proteic hormone to activate it. This is directly activated by the fact that cells, a different group of cells, can express different quality of steroid metabolizing enzyme. This makes that locally you have a different production of steroids, which is dependent from the genes activated by the singular cell. Then the intracerebral steroid level depend from that local production to general output and the intracerebral metabolism. What are the mechanisms by which sex steroids impact on the brain? First, I would like to start with the organization. As you know, we are all born with a female brain. But women, we have a transformation of our female brain in a male brain, which is dependent from the testosterone production from the testes of the fetus. The testes of the fetus producing testosterone modify the structure of the brain. And this is reinforced at the 3rd, 4th months after birth by that time where we have a small pubertal activation who bring an further increase of testosterone just after birth. Then steroids have an activational effect. They are very important to modulate the neurotransmitter function, to activate the receptor subtype crosstalk. They're able to induce receptor, and they have specific genomic and membrane effects on all kinds of cells living in the brain. They are touching the oxidative metabolism, they are responsible for the control of apoptosis, and they have specific cellular effects on neurons and on glial cells. They are able to produce the expression of synapses, they can control inflammation, and they have a very profound neurovascular effect. What are the non-reproductive central nervous system function impacted by sex steroids? You can see how they are important, mostly if you make the difference between humans and other primates. The affective state, mood, sleep, libido, sexuality, social behavior, the motor coordination, the neuronal excitability, the pain threshold, the auditory threshold, the cognitive function, and they have also multiple mechanisms for neuroprotection. When I have spoken about the CNS sexual dimorphism, this is dependent from the different expression of the various brain areas of estrogen, progesterone, and androgen receptor on their polymorphism and on different specific steroid metabolism who occur in the different nuclei. We have also structural sex difference who imply different neuronal subpopulation in brain areas. And this brings to what? This brings to a sexual orientation, to the gender identity, to the cognitive dimorphism, to the specific response of hormone therapy, and also to the different prevalence of neurological and psychiatric diseases. But you can see, speaking about estrogen, the most important estrogen, apart Estetrol, we will go back on that one, is estradiol in women. But you can see that women, they have very profound difference in their estradiol level. If you go from follicular, ovulatory, and luteal phase, where we have some 10 or some 100 picogram per ml, we go to the pregnancy where we have 1,000 until 40,000 picogram per ml, and for more than 30, 35 years, postmenopausal women, they go back to concentration of 10, 20 picogram per ml. How we can believe that this can have effect, positive, negative, different according to their concentration in their life? And also, what we have seen is that the brain shows a specific vulnerability, which is dependent from the sex steroid withdrawal. After menopause, women, they have a reduction in the brain in the expression of CRH, of propomelanocortin-related peptide and vasopressin, and increases in central noradrenergic system, while they have a reduction in central dopamine, serotonin, acetylcholine activity, and also a reduction in allopregnanolone production and then in the GABAergic function. And this brings to what? This can bring to some signs and symptoms who are specifically dependent from the individual. This is the new concept of the individuality. Vulnerability depends from an individual fragility. And this can be associated to stimulation of symptoms such as irritability, anxiety, fear, depression, fatigue, psychosis, decreased seizure threshold, but also hot flashes and autonomic hyperactivity. And this depends to what? When we speak about the symptoms of the menopause that we have resumed in that review that we published on Nature, you can see here they have central nervous system, skin, mucosal and hair changes, changes in sexual function, in weight and metabolic changes, in urogenital system, and in musculoskeletal. But if we go to see the central nervous system, which are who clearly indicate the great sensitivity of the brain to the hormonal changes, they are vasomotor symptoms, but also sleep disruption, depression and anxiety, cognitive changes, and migraine. But all these depend from specific activity. The vasomotor symptom and the sleep disruption depend to the increased noradrenergic activity with a reduction of dopamine, serotonin, and important increases also of neurokinin, kisspeptin, norepinephrine, and cortisol. And if you go to see what are the causes of the mood and cognitive function changes and changes in neuroendocrine activity, still the imbalance between growing noradrenaline and reducing dopamine and serotonin, but also a reduction in GABAergic function, a reduction in beta endorphin, in allopregnanolone, in all estrogen and androgen, and an increase in cortisol and increase in amyloid B. And this brings to the fact that if you look what happened in the different biomarkers in peri, and post-menopause, you can see here that we have selective changes in biomarker production, such as an increase in ATP, an increase in amyloid, changes in cerebral blood flow, changes growing of gray matter, why we have a reduction in white matter volume and in fractional anisotropy. And we have a reduction in glucose, cerebral glucose metabolism. All these indicate that these biomarkers indicate that we have a central general adaptation to the lack of steroids happening with the menopause. And one of the major chapters of our interest, which is associated to the fact that it is a huge health problem for health authorities, how to manage dementia in a growing population, is the fact that the observation that the prevalence of dementia is growing dramatically due to the aging population. All causes of dementia, different underlying pathophysiology, Alzheimer's disease, vascular dementia, there are many risk factors at that time. And when we see this in that nice cartoon showing in the left side the number of people with dementia in low and middle income countries compared to the high income countries, you can see here how in low and middle income countries the number are growing in a more faster way than in high income countries. And also, it is important to see what are the percentage of dementia touching the both genders, females and males. You can see here that females have a significant increase in the fraction of demented people in ages going above 75 to 84, and even more over 85. And as you know, the longevity of women is even bigger than men. Then this creates a huge and very important health problems. And very recently, it has been reinforced, the concept of the importance of the steroid depletion by that fantastic study that done by a friend of us, Walter Rocca in Mayo Clinic. They have two cohorts with bilateral oophorectomy, one of female with less than, who had bilateral oophorectomy at less than 55, and one who had an oophorectomy at 45 or less, who were followed for 14 years. And then the outcomes shows that the incidence, according to the time of the oophorectomy, you can see that overall mortality is significantly higher hazard ratio in those who had less than 45. The same is also for cardiovascular mortality, but also the same, very important, is cognitive impairment and dementia, that as early as be done an oophorectomy, as greater is the risk to develop dementia and other pathological processes. And this brings to the concept that the bilateral oophorectomy activate the aging process, activate the accumulation of senescent cells, the senescent cell-derived protein, the inflammation, the protein aggregation, mostly beta-amyloid, the mitochondrial dysfunction, the epigenetic alteration, the telomere attrition, the empiric signaling, the stem cell exhaustion, and this bring to a multimorbidity. The multimorbidity, I cannot go in details for time. They can touch the brain, they can touch the cardiovascular system, other somatic. I mentioned only the arthritis, for example, and also the development of diseases such as asthma, osteoporosis. But you have to take care of the brain, of depression, of anxiety, and all these who indicate the sensitivity of the brain, such as the cardiometabolic, the impact on diabetes, hypertension, hyperlipidemia, and all these stimulate the frailty and stimulate the reduced health span. Then we have a precise link between the damage done by early oophorectomy. And in 2002, it was published in JAMA, that very nice paper, in the same year when they published the WHI, very bad study, but in that study, they evaluate the incidence of Alzheimer's disease in the beautiful county of United States, the Cache County, in women and men. You can see the difference. Population going from 65 to 95, women in red, the men in yellow. It's a huge gender difference. But if the women, they were submitted to hormone replacement therapy, you can see what happened. In violet, the hormone replacement therapy non-user, then in green, less than 3 years, 3 to 10 years of treatment in blue, and in pink, treatment for more than 10 years. More than 10 year treatment cancel the gender difference in the incidence of the disease. And the same author, they went to have make a reanalysis and have make a longer follow-up of the study, evaluating the endogenous estrogen exposure, who was calculated talking the reproductive window in the age of the menarche to the age of the menopause, adjusted for pregnancy and breastfeeding. And they have found in that fantastic study that longer duration of endogenous estrogen is associated with better cognitive status in older adult women. And these effects are extended with hormone therapy use, particularly among the oldest women in our sample. And women who initiated hormone therapy earlier showed higher cognitive test score than those who initiated hormone therapy later, providing some support to the critical widow hypothesis of hormone therapy. And at the same time, a series of studies have shown that the treatment done in recently menopausal women don't change the cognition, but there were studies done for they have a short time duration. And in another study, Pauline Mackey and Victor Henderson, they publish that position statement from North American Menopause Society that estrogen may have positive cognitive benefits when initiated immediately after surgical menopause. Women with premature menopause are not well studied, but also they tell that hormone therapy in early natural postmenopause period has neutral effect on cognition, and hormone therapy cannot be recommended at any age to prevent or treat dementia. This was published in 16 and 17. But look, going on with the new investigation, Tommi Mikkola from Finland, he found that data on vascular dementia death and the exposure to hormone therapy. Please look the risk on your the right column in the exposure age of less than 5 years or more than 5 years for the vascular dementia, this is another chapter, the effect on the vessel, have a preventive effect at any age and for any duration. And if you go to look what kind of population was treated, if they were less than 60 or more than 60, you can see that either any hormone therapy or estradiol only or estradiol plus reversible, all of them, they show a very important protective effect on vascular dementia death, which were also the vascular effect of estrogen are important. If the same author went to evaluate the risk of Alzheimer, of death in Alzheimer's disease, you can see that there is a difference. This indicates only the longer treatment, more than 5 years, have a positive, have a reduction effect. And this happened mostly in younger individual and mostly younger individual and slightly less in above than 60. Then there is a difference in the response to hormone therapy according to vascular and Alzheimer, but Alzheimer dementia need a longer treatment. Alzheimer estrogen are able to influence both beta amyloid and tau proteins. This is something, biologically speaking, very important, with direct effect on neurons and action against neuroinflammation and oxidative stress. And but it's better and it's important to understand the early stage pathogenesis of Alzheimer's dementia. Is there a window for estrogen in Alzheimer's dementia? This remains the question. But when we go to see the amyloid deposition, and you imagine and you look at this beautiful picture who compare post-menopause, menopause, peri, and perimenopause, you can see here that human menopause is a dynamic neurological transition that significantly impacts brain structure, connectivity, and metabolic profile during midlife endocrine aging of the female brain. This is important. When I speak, the female brain have to face the aging process with the damage, with the fact that the menopause really introduces a steroid withdrawal. And if we have a replacement, this is a beautiful study from Roberta Brinton Diaz on the association between menopausal symptom therapy and the risk of neurodegenerative disease in general. Alzheimer's disease, Parkinson's disease, dementia, multiple sclerosis, amyotrophic lateral sclerosis, you can see in population who had oral or transdermal therapy in comparison to control. Please look how significantly, independently, and from the way of administration, oral or transdermal, stronger by oral way, all the neurodegenerative diseases are touched by the treatment. Then the treatment reduces the incidence of neurodegenerative diseases. And the result of the study was the use of hormone therapy associated with significantly reduced risk. Hormone therapy with natural steroids, everything beta estradiol or progesterone, were associated with greater reduction in NDD risk. Oral hormone therapy users show a significantly reduced for combined NDD compared to non-hormone therapy user, and the relative risk for transdermal hormone therapy user were significantly decreased for all causes of dementia and multiple sclerosis, and the greater reduction in risk of NDD, Alzheimer's dementia emerged in patients aged 65 or older. And protective effect on long-term therapy, more than 1 year. We believe now the treatment have to be done for 20 years. On combined NDD, AD, PD, and dementia was greater compared to the short-term therapy. But then now I would like to finish with some data that we have produced about the Estetrol. Estetrol, it was discovered many years ago. I was working on that at the end of the 60s and beginning of 70s. Then I went back later on when Jean Michel Foidart offered me the possibility to study the effect of Estetrol on the brain in animal models. And as you can see here, Estetrol have this peculiarity to be produced by the fetus and to have high concentration in the fetus, where you have to define the fetus from the very high estrogen exposure, and also high concentration in the mother. But what does the human fetus, why produce estetrol? I think that we have to go back to Aristotle. Nature does nothing in vain. Certainly, we have to discover the precise role of estetrol. But then, looking what is to the pharmacokinetic data, you can see here that they have a characteristic to have a very long half-life. Then you have a very maintained the exposition, and they remain a long time in peripheral circulation, who providing an easier way of action. And we have evaluated the effect of estetrol on the production of allopregnanolone. Allopregnanolone is a progesterone metabolite, which is important for its effect on GABAergic system. It's the most important endogenous antidepressant. And estrogen affects the allopregnanolone production by inducing the enzyme responsible of the metabolization of progesterone to allopregnanolone. And we have evaluated in various brain areas. And you can see here, this is the comparison in castrated rats of the effect of estradiol valerate 10 micrograms or 50 micrograms in different brain areas. This is frontal cortex and parietal cortex, and 1 milligram or 5 milligram per kilo per day of estetrol. You can see that given that estetrol have the same effect on the brain, stimulating the production of allopregnanolone in frontal and parietal cortex, stimulating also with a stronger action in the hypothalamus and in the hippocampus, then it maintains this effect, and also in the anterior pituitary and in peripheral circulation. Then estetrol have maintained this profound effect to stimulate the production of the most important GABA-A agonist steroid. And also in another area where our lab was working in the last 20 years is the production of endogenous opioids. And also for the endogenous opioids, this is the comparison between, in castrated rats, the effect of estradiol valerate 10 or 50 micrograms or 1 and 5 milligram of estetrol on the production of beta endorphin. And you can see here that also for beta endorphin, either in the hypothalamus as well in neurointermediate lobe, estetrol have a potent stimulator. And this is in the anterior pituitary, and in plasma you can see the different, they have a different effect in comparison to estradiol in the pituitary, but in peripheral circulation we have an increase of beta endorphin after estetrol administration as it was for estradiol. Then I think, ladies and gentlemen, what is our conclusion today? I would like to bring the slide that we recently published on Human Reproduction Update on our vision of hormone replacement therapy in women. And I would like to put in the chapter also the great importance to see the data that are now appearing on the effect of estetrol as estrogen. In healthy women or in women with early menopause, we have to start the treatment, and the therapeutic options are such that the treatment start with estroprogestin sequential or continuous oral, transdermal or transcutaneous, and different also other therapies. They have to be revised after 10 years, not stopped, revised, readapted, titrated to the needs of the patient, and she can continue also going on until very, very old age. But in women, we have already risk factor and comorbidities. Then we have to evaluate the proper treatment. And the proper treatment is to go according to the nature, not to go oral treatment, but to go, if possibly, to go transdermal or transcutaneous treatment with estrogens and also with the transvaginal progesterone to permit the treatment, and to go in, and also in this patient 10 years after, to make a recheck, a reevaluation, and to evaluate the possibility to continue the treatment. The possibility that estetrol offers now, by its lack of effect on the liver, is the fact that also in women with different comorbidities, we can maintain oral treatment if we will have from clinical results and for the result of the studies going on now, a positive, we maintain the vision of that absence of effect of estetrol on the activation of clotting factor by the liver. Then, ladies and gentlemen, I think that we have now new concept about menopause and aging. We have to rewrite the dynamics of aging process. And we have to remind that to maintain steroids mean to maintain the course of nature. And changing is different than aging. But I have also to remind you that no data indicate that in women who well tolerate menopause hormone therapy, we have to stop it. We have to adapt it, and we have to try to make treatment to protect the humans from that terrible incidence of dementia and Alzheimer who are touching our population in a time where we have no more the possibility to take care of ourselves, and the disability in elderly is a terrible problem. We have to prevent that one. And I think that in the prevention, Estetrol can have a very important word. Thank you, Jean Michel. Thank you, ladies and gentlemen. I'm sorry, you know, for COVID, I was obliged to remain at home. My heart is very close to you, and I thank you very much for your attention.
Thank you very much. Thank you very much for this wonderful talk. We appreciate your effort, and now we're going to ask for questions from the room here, if there are questions. Yes?
Professor Christos Sotiriou.
Can you hear me?
Yes, Professor Sotiriou, yes.
Thank you. I don't know if you can see me anyway. Can you hear me? So thank you very much for this great presentation. I'm being medical oncologist and treating breast cancer. So you didn't say anything about you know, potential contraindications of hormone replacement therapy for, you know, for postmenopausal patients, women and breast cancer. Do you have any comment on that? And if there is any difference according to estradiol replacement therapy or estradiol, you know, the first time that I heard today.
Thank you. Thank you very much. This is the most important one of the questions, probably one of the most important, because women, they have a specific fear about breast cancer. It touched the woman, not only for health problems, but also it touched the woman for their total body image and femininity. You know, certainly, as you know, we had several studies showing that it's not so much estrogens who have a greater importance in the breast cancer, but much the progestogen, And all the treatments done until now with estradiol only, they have indicated that the incidence of breast cancer don't increase. There are also some studies indicating that if you treat women only with estrogen, you can have also a reduction of the normal expected incidence of breast cancer. And also it has been shown that we have to use the natural progesterone, the progesterone or dihydrogesterone, which is the oral way of the stereoisomer of progesterone, not the progestin. The progestin, they're bringing with themselves some biological effects originating from their originating molecule, then testosterone, 7-methylhydroxyprogesterone, and by that way, they can negatively affect the breast, and they can increase the incidence of breast cancer. Then I think we have to reanalyze all the data and go back. Now with Estetrol, Professor Foidart knows, we have that probably new aspect that Estetrol have less promoting is even better, but we expect the data even better than estradiol. But I would like to go on the general concept. We don't have any data showing that hormone replacement therapy causes an increase of breast cancer if you use the proper way of administration, which should be probably transdermal, or if you use the proper progestin, which is progesterone, the natural progesterone or dihydrogesterone.
Before yes, do you want to ask something else?
Another question, maybe a naive question, because we are, you know, sometimes we induce ovarian ablation for our breast cancer patients for 5 years at least. So you say that, you know, regarding dimension, regarding you know, central neural effects, are those reversible, and how long it takes actually to have this kind of problems when you yeah.
I thank you very much for that question. I thank you very much for that question. The limit of 5 years, it was never demonstrated to be real. It was the people, they were afraid in general by the data. But now we have seen that the proper treatment, mostly for the brain protection, but also for the cardiovascular protection, is to go above the 5 years, to go from 5 to 10 and from 10 to 15. And also we have seen that the women who are still maintain positive effect and don't have any adverse effect by the hormone therapy, they have to reduce the total amount, but they can also continue until their 8th and 9th decade of their life. It's not needed to stop.
Thank you very much. Professor Casimir.
Yes, thank you very much for your presentation. I have also a question because you show us the very impressive effect on the prevention of psychiatric troubles and disease in women. Why estrogen didn't have use as a treatment or try as a treatment in psychiatric disease?
I thank you, Professor Casimir. Fantastic question, also that one. You know, first of all, all the doctors, apart of the gynecology, are a little bit afraid by the use of hormone, also because the hormone, they can induce uterine bleeding. And if you use only estrogen, you know, this can happen. And how to manage uterine bleeding if you are a psychiatrist is always something too much difficult. This was my first observation. But also it is important to mention that estrogen, we are speaking about the estrogen withdrawal, then the lack of steroids in a brain which is well adapted to have steroids always present at different concentration. Certainly, the data with the long-term therapy with steroid after menopause, they are different in comparison to the use of estrogen in women who develop a psychiatric disorder such as severe depression during their fertile life. These are depending from specific vulnerability and specific characteristics of the female brain of that woman, where serotonin normally may have a reduction in serotonin production, an imbalance, and reduction also in endogenous allopregnanolone production. Then I don't think that we can consider steroid as a treatment for a psychiatric disease, but we can consider treatment as a protection for vulnerable women who can develop psychiatric symptoms after steroid withdrawal.
Michel.
Thank you very much, Andrea, for this very nice lecture while you are sick with your COVID and you couldn't be with us. I wanted really to warmly thank you for this very elegant presentation at the time, which is very important for Mitra. And I see in the audience Graham Dixon, our Chief Scientific Officer at Mitra, who has developed with Mojos and the co-workers a very large comprehensive program on the neurological application of E4, including first in vitro studies on cells, on neurons and glial cells, but also in vivo in animals and also imaging in humans. And this lectures on the female brain is extremely beneficial for the audience, but also for the company. And I must say that I'm a little frustrated presentation because only women will benefit from estrogen. And what about the males? We don't produce estrogen, but our brain is still very competitive, no? And thank you very much for your lecture.
Thank you very much, Jean Michel. I am so sorry, and my wife also, she's so sorry that we were waiting to join you and Marguerite and to be with you and with Mitra friends in Brussels. I hope that, I'm sure that in Florence we can discuss all about it, and we are waiting the new data that you are going on studying the effect of Estetrol in postmenopausal women treatment. I think this is of major importance. And also, I think it should be of great importance also to have an assessment in the use of hormone replacement, in the use of Estetrol in contraception. As you know, in contraception, they were appearing data from some Swedish colleague about the suicidal risk in younger population in the first months of treatment in general. And I think that Estetrol, the combination with drospirenone, who have no effect on endogenous inhibition of endogenous allopregnanolone production, and the effect of the addition of Estetrol can be also considered in view of a protection, specific protection of this association in comparison to the effect of other contraceptives.
Professor Arnault.
I'm just an urologist, so it's a very simple question. How does the amount of estrogen receptors evaluates in women after postmenopausal women, as well as men, of course, but is there a decrease in the Mer receptors? Is there, after menopause, a decrease in the...
I'm sorry, I don't hear the question. I don't hear the question.
Microphone was not on.
Of course. My question is, with aging and in postmenopausal women, how does the amount of brain estrogens evaluate? Does it decrease? Is it stable? Does it increase? As well as the amount of estrogen receptors?
Thank you very much. Beautiful question. Okay, in the aging evolution, the total content of central estrogen is slightly progressively declining. The brain is not the same reduction as in peripheral circulation, because, you know, we have also a local estrogen production, which is partially maintained, but the mechanism is going down and the total content is reducing also in the even, so they have some difference in the various areas, but it is declining. And related to the incidence of steroid receptor in general, this is a different evaluation that have to be evaluated in a different way, have to be evaluated according to the different area. You know, the brain is a fantastic environment where you don't have the same concentration as in the muscle, as in the heart, in the different tissues. According to the different nuclear structure, you have a different local expression of steroidogenetic enzyme or steroid metabolizing enzyme. And this make that all the area who produces aromatase, you have a huge concentration local of that estrogen, while in a closer group of cells who don't produce aromatase, you don't have this one at all. Then it's important to evaluate the concentration area per area. But also with that way, a series of studies have shown that we have a reduction of receptors, a reduction also of the receptor for gonadal steroid. But this is sensitive to the administration of the steroids. As I remind you, the intracerebral content is depending from the flow coming from the periphery together with the local production. Then by the flow coming from the periphery, you can interact in the activation of steroid receptor. Estrogens, they can modulate, they can stimulate the production also of their own receptor.
No desensitization. No desensitization.
Yes.
No desensitization.
No, it's not apparent. The desensitization program is not apparent. You know, there are only a few studies. I think it's an area which has been until now poorly evaluated. I agree with you.
Thank you. We have a question from the audience, the Zoom audience. So Jean-Pierre Brion will ask a question to you. He's a member of the Académie, Professor.
Yes.
Hello, Professor Constantinescu. Thank you very much for the... Do you hear me?
Yeah. Yes, I hear you. I hear you.
Thank you. A beautiful review of, especially on aging and the relationship with Alzheimer's disease. As you nicely did show, initially it was a bit controversial, the effect of estrogen on the frequency of Alzheimer's disease. It seems to be more documented now. But you mentioned the potential effect of windows that would be therapeutically more interesting. Would you comment on the cellular effect of that? Is there anything in the hypothalamus, for instance, a difference or?
Yes, there are some studies who shows that the effect that we have to differentiate the study in humans as in animals. In humans, it is more clear that the administration in early stage of the menopause are associated to a better response of the individual, either with the different biological and also clinical evaluation. Then we have to start as early as possible, and the results indicate that as early you start, as better. But this is not true that if you start later, you don't have any results. You have results, but clinically speaking, of less importance in comparison to the other. Now, when we go to the animal, we have also to start with a reflection, a general reflection. The animal, they don't have the menopause. Normally, their life is stopped at the end of the possibility of the extent of their reproductive life. Only some whales have the menopause as women. Then the fact when we make the castration and we make the treatment, we make a picture who can be compared to the menopause. But this is quite different because this indicates that in the animal, you have a brain which is organized not to face that phenomenon. In humans, we have that prolongation of our lives. Our life now is 35 years more after the menopause is still existing, and we are still growing and probably will go on. And then for that one, it seems to be more specific, the protection, the concept of the window. Then as early you start, as better you have your effect. But no data shows that if you start later, you don't have any effect. You have minor effects.
Okay, thank you. Thank you.
No, it's my pleasure. Thank you.
Do we have any other question here in the audience? No, and we don't have any more questions on internet. Professore, lo ringraziamo, penso che la giornata è finita, adesso si può andare a riposare.
I will remain with you. It's so beautiful to be together, even though with that distance. I thank you very much, Jean Michel. I was honored by that invitation, you can imagine.
No, you can still come, Professor. This is not a problem for us. You know, here in Belgium, you're always welcome. So there's no need of a conference.
Thank you very much.
Thank you so much. So let's now see how Estetrol, a native estrogen, has become an oral contraceptive method. Mr. President, you will introduce the third speaker of the day.
Thank you. So I'm greatly honored. It's a distinct pleasure to introduce Professor Michel Cranin. Michel Cranin is tenured professor and director of family planning in the Department of Obstetrics and Gynecology at the University of California, Davis. He did his undergraduate studies and medical studies at the Northwestern University in Chicago. He received an MD in 1988. He then went for training as resident at the University of California, San Francisco, became an assistant professor, and then moved to University of Pittsburgh, and then in 2011 to the University of California, Davis. Dr. Cranin is a well-known expert that receives research funding from the NIH, USAID, WHO, and CDC, as well as private foundation and industry, and that has enabled him to be at the forefront of discoveries in female and male contraceptive development, including medical and safety oversight of national and international trials. He is an author of more than 300 peer-reviewed papers, review articles, book chapters, and books related to family planning and contraception, especially most recently the 6th edition of Sperhoff and Darney's Clinical Guide to Contraception. He is also past president of the Society of Family Planning. And with that, we really thank you very much for being with us, coming from so far away, and look forward to your lecture.
Thank you very much, and thank you very much for this honor to present at the Royal Academy, and obviously this is to honor Jean Michel Foidart, and I appreciate that you wanted to include me. I'm a clinical researcher, so listening to all of you throughout the morning has been a great lesson for me, as it always is to work with Jean Michel. I learn something new every day. But my focus is more on how you take these ideas and these discoveries and run clinical trials and prove what happens when we use these products in humans. So I was asked to review a little bit of the data on Estetrol in contraceptives, which is days of data that we're going to try to break down into 20 minutes. These are my disclosures that I do work with various companies. I always like to say that I work with everyone, so I am not biased by anyone. We're going to learn today important outcomes from phase 2 and 3 clinical studies of Estetrol in combined oral contraceptives. I'm going to try to focus on what I feel are the most important areas and how the phase 2 studies taught us more about understanding the impact of combined oral contraceptives on hemostasis and how Professor Jean Michel Foidart has created a lasting legacy. This development program dates back more than 15 years to early looks at Estetrol in clinical use, but more importantly, things started to move along as phase 2 studies with looking at Estetrol in combination with different progestins at different doses to try to find the best combination for use in oral contraceptives. Importantly, Estetrol was looked at with both levonorgestrel and drospirenone to look at bleeding patterns, ovarian inhibition, metabolic, hemostatic, and other endocrine outcomes to try to find the ideal formulation. I get asked very frequently, why was this product combined with drospirenone? Because it worked the best when it comes to bleeding profiles and ovarian inhibition. And this is what led to the phase 3 program with the use of Estetrol and drospirenone in 2 parallel large clinical trials, which I had the pleasure of working with Professor Foidart and the team at Mithra in bringing this all to fruition. When we look at the phase 2 program, the data on bleeding and ovarian inhibition, to be fair, isn't as important right now because we have phase 3 data. But I think the data on hemostasis has really been eye-opening and very important. As someone who has been working my entire career on contraceptive development within the female contraceptive world, the goal has been, how do we provide something that is either estrogen-free or contains an estrogen that doesn't increase the risk of venous thromboembolic disease? For a young, healthy woman who's using an oral contraceptive, going through a venous thromboembolic event is life-changing, right? This is potentially very morbid or could even kill her for somebody who is an otherwise healthy user. This is the downside of combined oral contraception, despite all the benefits. So how can we find something that could be safer and provide less risk, especially where I live in the United States, where you see increasing amounts of obesity and even increasing risks. These are the likelihood of developing a venous thromboembolic event, and we feel comfortable using estrogen-containing contraceptives, even though the risk is increased from a non-pregnant, non-combined oral contraceptive user, because the risk is so high in pregnancy, and importantly, it's really highest in that postpartum period. Now, recent concepts on trying to understand combined oral contraceptives and both the estrogen and progestin impact over the last 20 years, I think have really fell short. We didn't have data to try to understand it. We just presumed that we always have known that the estrogen increases the risk. But the data since the early 1990s has suggested that the progestin has an impact, and I've lived through my career developing in a world where people thought that the progestin actually also could cause venous thromboembolic disease. Newer progestins actually cause this process. But we really lacked the data. We didn't understand the balance between procoagulant and anticoagulant factors and how the estrogens and progestins really affected these differently, primarily because we only had one estrogen. We always had ethanoestradiol, which is in more than 99% of combined oral contraceptives. So whenever we were altering the product, it was the progestin we were altering, but kept the estrogen the same. So it was easy to try to blame the progestin when we saw different rates in clinical trials or in large population-based studies. The regulatory agencies look at this differently on the different sides of the ocean. The Food and Drug Administration just has labeling guidance and wants all the labels to say the same thing, whereas the European Medicine Agency wants actual testing of different outcomes without really knowing what's important. It just says, here's the panel of tests, we want you to run these tests as part of your phase 2 program, but we have yet until recent data to truly understand perhaps which one of these or which parts of these studies might be most important. It's been really important, though, as we look at all of this information to understand that these tests are not necessarily validated predictive markers. We don't know which ones are most important, but we now have some more clues about what may happen as we use different estrogens and different progestins in clinical trials. And the phase 2 hemostasis study during the development of the estetrol drospirenone contraceptive is really what gave us that first clue. This was a randomized open label study with 2 different comparators, an ethinylestradiol product with levonorgestrel and an ethinylestradiol product with drospirenone. So this, for the first time, gave us the ability to test a product that had the same progestin, drospirenone 3 milligrams daily, with 2 different estrogens. So we could try to differentiate what was related to the progestin and what's related to the estrogen. And these were reproductive age, healthy, non-obese women. They had baseline coagulation testing and then treated daily for up to 6 months with testing at 3 and 6 months to look at changes over time. Now, this is a busy slide, and those of you in the back of the room are actually in the best place to look at this, because this is take the general overview look. The green bars are the ethinylestradiol drospirenone, and the blue bars are estetrol drospirenone. And what you can see, no matter what type of factors you look at, is that the green bars cause more of an undesired outcome than the blue bars. And the only difference here is the estrogen. Same progestin, different estrogen. So for the first time, we really can understand, is it the progestin that makes things worse or not? No. It's the progestin will modulate the effects of the estrogen. And it is really for the first time, our ability to see these differences has shed things with better light. And I want to focus on the anticoagulant proteins, because I think when I look at this data, this is the most important changes we see. So the ethinylestradiol levonorgestrel product is heralded by the different regulatory agencies around the world as the one that they consider to have potentially the lowest clinical risk, or have had the lowest clinical risk. So the pink bars give you that potentially lowest clinical risk product as a comparator. What you can see is, especially as you look at protein S, the dramatic difference when you have drospirenone being exactly the same between ethinylestradiol and estetrol on the outcomes of anticoagulant proteins, that it is all driven by the estrogen. When I look at this, I think that I see that drospirenone, compared to levonorgestrel, drospirenone is a very neutral progestin. It doesn't have as much impact on the liver, on the kidney. That's a whole other lecture. But what we're getting a chance to see is the full effect of the estrogen. And what we see here is that estetrol, with drospirenone, you have minimal impact from estetrol, but you have a dramatic impact from ethinylestradiol. And drospirenone isn't inhibiting that, whereas levonorgestrel may be inhibiting the full effect of ethinylestradiol. So it's not that the progestin induces risk, but it modulates the risk of the estrogen. All right. Another way to look at this is with activated protein C. We've known for decades that APC resistance is a major risk factor, especially for those who carry a genetic predisposition related to APC resistance. And it's a promising biologic marker, but only in recently, again with studies comparing estetrol and ethinylestradiol with the same progestin, we've been able to see how this works. So this is activated protein C resistance graph that shows coagulation or thrombin production over time. So this is in a patient that with no activated protein C with the plasma, so you can see the normal formation of clot. And when we put in activated protein C, you can see that this lower line gives you the less thrombin formation, less clot formation, because you resist the clot formation. When you add an ethinylestradiol containing combined oral contraceptive, whether it be with levonorgestrel or with drospirenone, even in the presence of activated protein C, you see more clot formation. So these products are inducing clot. Well, what happens when you add ethinyl estradiol with drospirenone? There is some induction, but significantly less as compared to these other products. And notice that the highest dashed line here and the lower blue dashed line have the same progestin. The difference is the estrogen. So you have less impact of estetrol on clot production than you do compared to ethinyl estradiol, and the progestin isn't causing this. It's all a modulation of the estrogen risk. So you can take those results from this phase 2 study, and based on predictive modeling that is held up pretty well when you look at real-life outcomes, you can see the different prediction of VTE risk with different estrogen-progestin combinations, and you can see where estetrol drospirenone will fit to potentially have the lowest risk of any combined oral contraceptive. This is all theoretical, and we need phase 4 studies to really, phase 4 human studies to really bear this out. So estetrol drospirenone from the phase 2 studies, to me, the most important thing we learned is it has less pronounced hemostatic effects than an ethinyl estradiol drospirenone product, that they were comparable or better to ethinyl estradiol levonorgestrel products, and that the decreased hemostatic effect when you hold the progestin the same underscores the importance of the estrogen type in combined oral contraception. And as I alluded to earlier, we need large phase 4 studies to really confirm this. Well, what clinical data do we have to try to understand these outcomes before we have phase 4 studies or until we have phase 4 studies? Well, obviously the phase 3 studies, which were performed for efficacy, safety, and bleeding profiles, were really great studies and will give us the first view of that. So let's review a little bit of the phase 3 data, also to look at efficacy and other outcomes. These were parallel studies done in both the US and Canada as one study, and the European Union and Russia as another study. I don't know today if it would include Russia, but this is the data we have. The primary objective was to evaluate the efficacy of this combined oral contraceptive, and secondary objectives were to look at safety and bleeding profiles. Now, the populations were very different. I come from a different land than you all live in. The patients I see are different than the patients you see, and the patients that enroll in clinical trials are different. So as we look at outcomes, we need to importantly understand some of these differences. There are major differences, and I'm going to focus just on the 16 to 35 or 18 to 35-year-old population, because that's the main population looked at for efficacy. The 10% of people enrolled that included that were 36 to 50 are for safety and bleeding outcomes as well, but the primary efficacy population is women up to the age of 35. And you can see dramatic differences in race, you can see dramatic differences in BMI, and that I live in a world where almost 25% of the study participants were obese, whereas in Europe and Russia, 5%, again, very different patient population. And the types of products they were using as they entered the study were different, where 60% of people in the European study were already using a pill. They were used to using it, they were just switching to a new pill in the study, whereas that was only about a third in the US. So the mix of patients entering the study is very different. And the outcomes then would be expected to be a little bit different. The Pearl Index, or the primary measure of pregnancy, was 0.5, or roughly a pregnancy rate of 0.5% in the European study, and 2.5% in the US trial. It's just for those of you that don't work in this world, those are both very highly effective numbers. The European study, more effective, primarily because of the patients that were enrolled, and US women, from study after study, shows US women tend to be, have less adherence when they enroll in pill studies. But a Pearl Index of 2.5 is right in the range of what would be expected for a contemporary combined oral contraceptive. So bottom line is this pill works as expected. All right. Importantly, in the US trial, because there was such a high proportion of obese users, it gave us the opportunity to look at, was a difference, did a difference exist in obese and non-obese users? And importantly, you can see the Pearl Index was the same, 2.6 and 2.9, not only statistically the same, but clinically that's the same, with an overall contraceptive efficacy using life table analysis of 98%. So this is a highly effective combined oral contraceptive. It works the way we expect. What about bleeding? I have a colleague in the US who kept telling me, this is a weak estrogen. You're really just giving a progestin. It's a progestin-only contraceptive. You're going to see bleeding. This doesn't work. This is an amazing product. And again, the back of the room, you can see this best. This is the bleeding profile in both the European study and the US study. Importantly, red is bleeding and blue is spotting. What you can see really clearly is in this regimen that was given with the idea of causing cyclic bleeding is that participants experience cyclic bleeding. So it works as a really good combination of estrogen and progestin. There's a good balanced effect on the endometrium, so you get bleeding when you want it. You do get some unscheduled bleeding early on, as you would expect with any combined oral contraceptive, and over time, the unscheduled bleeding between the scheduled bleeding episodes goes down. And importantly, what you in the back can really see well is that almost all of that is blue. Very little of it is red, because the vast majority of bleeding that occurs is spotting. Spotting was defined as needing no sanitary protection, no menstrual protection, product protection, so that the majority of women who experience unscheduled bleeding experience something that they weren't even wearing protection for, and that it goes down over time. This is a product that has a great bleeding profile. You can see the unscheduled bleeding rates go down over time, and that the majority of the unscheduled bleeding is spotting, such that only 3% of women over an entire year discontinued because they didn't like the bleeding. And then lastly, let's look at safety. A very neutral impact on lipids, cholesterol, as we would expect with this product, and triglycerides. And this is really important. Ethinyl estradiol products almost universally increase triglycerides, which in young women is important, especially in obese young women, and we don't see that increase with an Estetrol product. We see no change in glucose metabolism or in long-term glucose outcomes, with hemoglobin A1C staying pretty steady at about 5.3%. When we looked at adverse events that led to discontinuation, to me that's always the key of safety. What kind of experiences were women having that they said, I want to stop this product because I don't like it, because it's causing a side effect I don't like? The regulatory agencies want the labels to include any outcome that is occurring at a rate of 1% or greater. For this product, there was just one, and that's bleeding irregularity. Any other complaint that led to discontinuation occurred at a rate less than 1%, which is almost unheard of with most of the combined oral contraceptives on the market. And you can see these reasons here, things that people will complain of with use of a combined oral contraceptive, but at such low rates that they were not causing high rates of discontinuation. And you can see at the bottom right here, one VTE event occurred during the study, and that's important. It occurred in the European trial, not in the US trial, where we might have expected it since we have so much more obesity. So I want to refocus on that. We talked earlier about these risks of developing a VTE event, and we had phase 2 data, small phase 2 studies that look at coagulation parameters. We need population-based phase 4 studies to ensure what the real rates are. But these phase 3 trials with thousands and thousands of women were our first chance to look at, in some type of large population, what happens from a VTE event standpoint. So I'm going to focus on some US studies because of our obesity rate, where we're more likely to see VTE events. These are contemporarily approved combined hormonal contraceptives. You can see we have a 10 microgram ethinyl estradiol product with norethindrone acetate, an ethinyl estradiol sugesterone acetate vaginal ring in the US called Annovera, and a new transdermal patch recently approved in the United States called Twirla. These phase 3 studies had varying rates of obesity, but you can see across the board in phase 3 trials in the United States, in that population, we see about 3 to 4 VTE events in every phase 3 trial. That's what we expect. It's an unfortunate outcome, but with use of ethinyl estradiol products, we see 3 to 4 events, no matter the dose, even with this extremely low 10 microgram dose. Well, what did we see in our studies of Estetrol? In the US population with 23% obesity, we saw 0 VTE events, and there was 1 in the European trial, not the 3 to 4 we typically see. Again, we need population-based studies to know how this truly translates, but in the first translation of the phase 2 data to phase 3, where we have thousands of women, we can see the beginning of what I call hope. That hope that we may have an estrogen that really does translate from the bench to the bedside in something that can provide lower risk for women who want to use a combined oral contraceptive. So this program, in summary, has allowed us to understand we have a product that has high efficacy, an excellent bleeding profile, excellent cycle control, good tolerability, very low rates of discontinuation for adverse events. And in my career in contraceptive development, I think this is a game changer. So thank you for allowing me to speak to you, to try to make everything as brief as possible. And thank you for my friendship and the continuous learning I get from Jean Michel Foidart, who is a great inspiration to all of us. So thank you.
Thank you very much for this very, very interesting presentation. And I'm opening it to questions, first of all, from the room. Are there any questions? I had a question, but of course, probably your patients are not eligible that would have also some myeloproliferative neoplasm or clonal hematopoiesis of indeterminate potential. Do you have any experience of people that have, by chance, a couple % of driver mutations for hematologic cancer that are prone to this type of thromboembolic events, and were they, by chance, on therapy in the trials? No.
I think you know the answer. No, those people would be excluded from the studies. And I think the important thing is, until we have more data, those type of patients really should not be using estrogen of any kind until we know the safety. But what we're looking at is how do we, over the next few decades, drive the ability for patients who may need some type of cycle control and need an estrogen to have something that could be safer. But you know, the point first is and foremost is to have a product that's available for everyone, so patients like this would not be in any clinical trial or even still be recommended to use a product like this.
Exactly, but in the last years there was this discovery by Ben Ebert from Dana-Farber that there are people that have more than 2% clonality with mutations such as DNMT3A, TET2, and so on, and they were not known. They don't have any disease. They have absolutely nothing. So do you have DNA from these people in your clinical trials?
No, we don't have DNA.
Okay. Other questions?
Great question.
Yeah, Professor.
I think it also speaks, though, to the fact that we know that a lot of people have underlying genetic predisposition to coagulation, hypercoagulability, but it doesn't always translate clinically. So we still don't fully understand, just because you have a genetic risk, the permeability of that risk, and it varies from individual to individual.
Thank you very much for your beautiful lecture. I would like, if you have the opportunity, to test the smoking effect in your population.
So the question about the effect of smoking. The effect on what outcome?
Thromboembolism, or also on metabolic disease, metabolic disorders, of course.
Right. There is nothing from the trials that shows any difference in those who smoke. But to be fair, the smoking rate in the study, even for the European population, was relatively low. So low that the numbers were too small to really look at large differences in the outcome. Thank you. Which I herald as a great thing, because the fewer and fewer people that are smoking, the better we are as a whole. And I remember days when we could easily test this out because so many people did smoke. And it's nice that fewer and fewer people are smoking. In the US, they're smoking more marijuana, but not smoking as much cigarettes.
Really, really quiet Saturday for me, you know.
Thank you for allowing me to join you. I appreciate it.
Thank you. No, but wait a second, because maybe there are still questions here in the audience. I was just answering to Professor Constantinescu, who was asking me if I had questions on the web. And apparently no question for me. Is there any question here? No? May I ask a question? Which is not scientific at all. It is more, no, priority to someone who has a very good question. Stéphane Lourian also has a question, apparently, and I don't see him. Ok, donne-moi. So, d'abord dans la salle, on va vous donner la parole, et puis on viendra, je lirai la question du professeur Lourian.
Thank you very much for the presentation. That was an excellent overview. And I guess one of the questions that comes up is knowing some of the benefits that we're seeing with Estetrol compared to, can you hear it?
Yeah.
Some of the benefits that we potentially see with Estetrol compared to ethinyl estradiol, and translating that into clinical practice, I guess that brings up the question of preferential prescribing. So patients who may not have an absolute contraindication according to WHO or CDC, but maybe they are at an increased risk of certain outcomes. So would this be something we would preferentially be prescribing to that population as opposed to one of the other oral contraceptives? Recognizing that, to me, I agree, I think this is a bit of a game changer. So just wondering what your thoughts are. In particular, you talked about patients with a higher BMI. So is that someone who we might be using this for as opposed to another one? Because that's a big question we always get, right? Which one do you choose to start and why? And then do we run into issues of maybe seeing outcomes, adverse outcomes because we're preferentially prescribing? So just wondering what your thoughts are on that.
The idea is, do providers preferentially prescribe this, or could they preferentially prescribe this product to people that are higher risk and use the existing ethinyl estradiol products for people they perceive at lower risk? And I think that really comes in how we message this new product, that any ethinyl-containing product in any young, healthy woman can cause a VTE event. And should phase 4 data prove what this phase 2 and now phase 3 data strongly suggest, I would make the argument that anybody is at risk that you start on ethinyl estradiol. And I think you raised the point really that we need to be careful in our messaging not to preferentially prescribe. That it doesn't matter whether the woman is thin or thick or smokes or doesn't smoke, that if she uses a combined oral contraceptive, her risk of a bad outcome can occur regardless. And I think you really herald more that we need to be aware that that can happen. And I think it's important for the messaging.
Thank you. May I just ask one question? It's about dosage. Just for the women who are going to use this contraceptive method, I know that in the US you are used, a lot of women are used to get shots. Is it a possibility? And is it also a possibility maybe for menopaused women? Because I'm wondering if someone who is in this phase of her life wants to take a pill each morning, or if it is easier maybe to get an injection like once a year to solve the problem, I would say.
So for the use of estrogen in hormone therapy, it's typically in a pill or in a patch. Estrogens, they're too big and don't work well in an implant, or in an injection. You get too high levels to be maintained over time. But there is wonderful work going on with Estetrol for menopause therapy, for hormone therapy, as kind of was alluded to. But in hormone therapy, it's injection or patch. There also, or in a transdermal gel, like a lotion you rub on once a day.
Okay. Thank you for the conference. Thank you for the answer. And we will now explore the final chapter of the symposium, the relief of the postmenopausal symptoms with Estetrol. And Mr. President, you will introduce the last speaker, who comes from Belgium. He's the first one.
So it's a really great pleasure to introduce Professor Ulysse Gaspard, who is actually a member of our Academy since 1993 and membre honoraire avec droit de vote since 2013. He obtained his medical degree, Doctor en Médecine, in 1968, specialized in obstetrics and gynecology in 1972, also in nuclear medicine 1974, and obtained his agrégation de l'enseignement supérieur en sciences médicales in 1980 in Université de Liège. He has been the founder or a founding member of the Service of Gynecology in 1988, Professor ordinaire of Obstetrics and Gynecology, chef de service at Centre Universitaire Liège-Sartilleman from 1994 to 2007. He has also been at the Notre-Dame des Bruyères, the university center there. And then he has remained as a consultant in Liège, CHU, Brûle, from 2007 to 2021. If one looks in the PubMed, one sees more than 270 publications in reproductive endocrinology, metabolism, and clinical medicine, contraception, menopause, hyperandrogyny, and of course the use of SERMs and SPRMs. He is a founding member of several national and international societies and also a member of the editorial board of Revue Médicale de Liège. He is now a senior consultant with Mitra Pharmaceuticals Liège, and we are very happy to have you here, and we're looking forward to your lecture.
Dear colleagues, first of all, I would like to dedicate this presentation to my friend Jean Michel Foidart, with whom collaboration since numerous years is so fruitful and exquisitely enthusiastic. Thank you. Now, we are going to the chapter which follows contraception when we are entering menopause, and I would like to speak to you about efficacy and safety of Estetrol treatment on the full spectrum of menopausal syndrome versus non-hormonal neurokinin 3 receptor antagonists. My disclosure is very simple, as it has been said. I'm now a senior consultant for Mitra Pharmaceuticals after a long career as a professor of gynecology. Well, you know effectively very well now what is Estetrol. It's 4-hydroxyl. You also know from the previous presentations that E4, and it is very important, has a unique pharmacokinetic profile among estrogens, and particularly when you compare it to estradiol. A much greater bioavailability, a freer proportion in estetrol circulating in the plasma than it is the case for estradiol. Its metabolites are only glucuronate and sulfate conjugates, and it has no other metabolites, which is not the case for estradiol, with 2-hydroxy and 4-hydroxyl derivative that may be genotoxic and even carcinogenic, and which has a lot of correlation with the cytochrome P450 enzymes, so much drug-drug interaction, which is absolutely not the case for Estetrol. And last thing, very important, a long elimination rate of 20 hours, where estradiol has a very short half-life. All this is really important to consider in the activity. And you have seen also in the previous presentations how much tissue selective it can be. It has the same estrogenic activity as the other estrogens on the uterovaginal action, on the cardioprotective effects, on the prevention of bone problems, also a protection on brain, which has been shown by Andrea Genazzani. I would say he is very liver-friendly because he's much less effective than estradiol itself, particularly when you remember that hepatic coagulation factors are mainly formed in the liver, and which is not the case with Estetrol. Minimal impact on different liver proteins, and a minimal impact on the metabolism in general. So also on the breast, which has not been mentioned before, it is antagonistic on the effect particularly of estrogen, of estradiol itself. Estetrol has antagonistic effect on the breast cell proliferation, migration, and invasion. So this is also a difference which has to be studied much more later on. So the take-home message from that is unique pharmacokinetic and metabolic profile, unique mode of action, first native estrogen with selective tissue activity, which qualifies him to be interesting for an overall effect in menopausal hormone therapy. Well, what is the situation? You know that every woman is going to go through menopause, provided she lives over 50 years of age usually. And that means that many women will spend 40% of their lives in the postmenopause. In Europe, there are 75 million of postmenopausal women. And we had a scare in menopause after the Women's Health Initiative because they were tracking problems like thrombotic risk, like breast cancer risk, and so on. However, their fear were diminished with the longer follow-up about that after that study. And now the scientific associations have a positioning statement which says, well, benefits of HT are important, and the most effective treatment is by estrogens themselves. However, there is a need for new hormonal products that could enhance benefits and minimize risks, and this risk being essentially in terms of thrombotic risk and a smaller risk of breast cancer. So we were interested to develop another estrogen, and you have seen how unique it is. And the phase 1 was directed by Herjan Kunin-Benning, who in his phase 1 showed the proof of concept of Estetrol that could probably be used in terms of hormone therapy for menopause. And so we embarked into the study, phase 2B, which is a multicenter, randomized, dose-finding, double-blind, placebo-controlled trial, including 257 postmenopausal women, mean age 54, mean BMI 26, in 5 treatment groups with dose levels of Estetrol going from 2.5 to 15 milligram, and of course a placebo group for the comparison. It was a 12-week study with administration of A4 or placebo alone, and after that, well, you can't leave the endometrium without a protection, and after these 12 weeks, there were 2 weeks of administration of a progestin, in that case dihydrogesterone, just to protect the endometrium. The primary endpoints were the following. We selected postmenopausal women with at least 50 hot flashes per week. And then we looked for the change from baseline in frequency of moderate to severe VMS, vasomotor symptoms, that's hot flashes, at week 4 and week 12, compared, of course, to the placebo. The same for the severity, to assess the severity of these hot flashes. And the secondary endpoints, which are not minor ones, even if they are listed as secondary, is the action on the vaginal cytology, maturation value, genitourinary symptoms of menopause, and of course, the health-related quality of life which ensues. So, first, the reduction in the frequency of VMS is, of course, very well seen. I try to have the... Okay. Reduction here, and which is best with the 15 milligram Estetrol dose daily administered. And you see that at week 4 and at week 12 as well, we have a significant reduction versus the placebo, and also, of course, versus baseline. And we were very interested to have the first results of the phase 3, which shows a more robust way, because it's a very large group of postmenopausal women, that it is statistically very significant also. The same applies to the reduction of severity, which is with the 15 milligram dose of Estetrol, very significant versus placebo at 4 and 12 weeks. And this is also the case with the phase 3, as I have told you before. An excellent result was the responder analysis on the hot flush frequency. You see that on the left part of the graph, more than 90% of the women responded with more than 50% decrease in VMS frequency, and even better, almost 80% responded with more than 75% decrease, which is, comparing to other studies of other estrogens, a very good result. And you see here, the higher the column, the higher the estrogenization of the vaginal mucosa. And in that case, you see that already with the smallest dose, you have an increase in that maturation, which brings the superficial cells of the vagina, which are in a strategic position, to be very much estrogenized. And that means that with the 10 milligram or the 15 milligram, you reach an estrogenization that is the same that during a normal cycle in the follicular phase, which is an excellent goal to be attained. Of course, when you restore the vaginal maturation, well, you have much less genitourinary symptoms, and which are there, and they are scored from, Likert scale from 0 to 4. And you can see that one of these symptoms, very important, is the vaginal dryness, which is coming from the hypertrophy of the vagina at the time of the lack of estrogens at the menopause. And that with, once again, the 15 milligram dose, you have an effect which is significant versus the placebo. Note that for all these symptoms, there is a nice decrease versus baseline in terms of these symptoms. And one of the most important, the most bothersome, is vaginal pain associated with sexual activity. And you see that already with 5 milligram, but also with 10 and 15 milligram, we have a big statistical difference with the placebo also. The same applies with the nice reduction in the vaginal vulvar irritation and itching and in dysuria, but not to a point attaining significance for the placebo, but it has a significance versus the baseline. So this is a restoration of the vaginal structure and of the vaginal function. Well, if you decrease hot flushes, at the same time you are decreasing many other things. And you increase the quality of life of that woman. And we stated that with the menopause rating scale, you have there 11 symptoms which are distributed into 3 domains: the physiological symptoms, the somatovegetative ones, and the urogenital. And you see once again with the yellow line that the one who alleviates more the problems and improves the quality of life in that way is the administration of 15 milligram per day. Sorry. Well, the slide doesn't want to come, is in fact with well, it's not that, there is no problem about that. It was just the one before. To show, no, she's not appearing. Okay, leave it, no. Okay, with 15 milligram of Estetrol, all these 11 symptoms that you see have been reduced at least by 50%, and this is impressively significant. So now, after showing that efficacy aims are attained, let's say something about safety. And as has been very well told to you by the previous presenter, what happens with other estrogens after the menopause is a decrease in the natural anticoagulants, let's see, protein C, free protein S, for instance, and an increase in the procoagulant, which happens. That is to the point that these administrations of other estrogens are associated with the risk of future thrombosis with an odds ratio ranging from 1.9 to 3.2 versus non-users. Okay, this is important to consider. Well, and this is a slide in the older patients that is really in phase with what happens in the younger one, that if you have the placebo in blue, and you have the administration of 15 milligram of Estetrol in red, you see that there is almost no change in any of the procoagulant and anticoagulant factors, and that there is only one single small increase here in activated protein C resistance, a very small one, which is interesting to interrogate more effectively. And you see that when you assess APC resistance here, you have a small increase in it, and you see it exactly also in the pill that Professor Krenin showed to you, because they contain both exactly the same amount of Estetrol, that is 15 milligram. And when you compare it with what happens with APC resistance for other pills containing ethinyl estradiol, which are twice, thrice more elevated, there is no, you cannot make any similarity between both of them. What is then the message which is important? The observed hemostasis changes in these older women with E4 can be considered as clinically non-significant, no change. Data on APC resistance support the hypothesis that Estetrol may be associated with a lower risk of thrombosis, and you have seen with Professor Krenin this fantastic correlation line between increase in APC resistance and increase in venous thrombosis with a R for correlation of 0.95, which is very impressive. Efficacy and safety profile, let's make a small summary. Well, it is effective, very effective on frequency and severity of hot flushes. No impact on coagulation factor, lipid, I had no time to get into it, and glucose metabolism parameters, potential beneficial changes in HDL-C, which is increased, in HbA1c, which is decreased, only minimal increase around 10% in triglycerides, which is not the case with other estrogens, which do increase it very much more. Reduced cross-lapse and osteocalcin values, indicating reduction in bone resorption. There is a good estrogen, in fact. Significant increase in the baseline SHBG, which is much less than with estradiol, for instance, showing less impact once again on the liver. No apparent safety concerns. Estrogen was Estetrol was well tolerated, increased endometrial thickness due to estrogen alone. Remember, 12 weeks with estrogen alone, but it normalized completely with the progestin for 14 days, and there was no hyperplasia before or after that progestin administration. Okay? Well, we were encouraged then to go into the phase 3 with more than 2,000 women, postmenopausal women, two randomized, double-blind studies versus placebo in this phase 3 for evaluation of the efficacy on the one side and safety on the other side. The first one of these was conducted mainly in Europe, and it assessed efficacy of 15 and 20 milligram of Estetrol in 600 postmenopausal women for a period of 12 weeks. And then, in another part of the study, to evaluate the endometrial impact of 20 milligram of Estetrol, plus, of course, 100 milligram of progesterone, of natural progesterone, for 1 year, and that was in 600 postmenopausal women also, and even a little bit more. And the other part of the study, the COMFORT 2, was run essentially in the USA and in Canada to assess in the same manner the efficacy of 15 and 20 milligram for 12 weeks in 600 menopausal women. And there is a big difference is to evaluate the general and endometrial safety of 15 and 20 milligram of Estetrol alone for 1 year on a specific and stringent demand of the FDA. That means that the endometrium is then in danger of any type of hyperplasia, but the FDA wanted to see up to which level you could happen, as you do with other progestin, other estrogens given alone for so long a period, of course, which can boost a level of hyperplasia and so on. So we were rather hesitating to do that, but we were obliged to do. Both studies, in terms of efficacy, all the studies are not terminated, so it's preliminary results. Both studies demonstrated a meaningful reduction in VMS frequency and severity from baseline and compared to placebo. And secondary endpoints evaluated at 3 months then only in the European study, because we have not yet the other results, suggest a positive impact of A4 on quality of life. That means on hot flashes, mood swings, anxiety, sleep, joint pain, skin and hair quality, and last but not least, sexual activity and libido. The take-home message is that E4 is effective for the treatment of the full spectrum of menopausal symptoms. Then I would like to tell you some words about neurokinin 3 receptor antagonist used or which are going to be used for the treatment of hot flashes. For that, we have to go inside the hypothalamus, and then you see an interesting nucleus, which is called in the human the infundibular nucleus, and in those who are used to working with animals, the arcuate nucleus is the same. Huh? And among other things, it contains a neuropeptide, which is kisspeptin, which is helping GnRH, gonadotrophin-releasing hormone, to act on the pituitary to deliver gonadotrophins to stimulate the ovary and to produce estrogens. And these estrogens have, during the sexual life of the woman, the reproductive life of women, a role which is mitigating the action of this nucleus. After this kisspeptin it is also containing neurokinin B and dynorphin, accessorily. But this neurokinin is capable through a primary receptor, which is the NK3 receptor, to bind to the median preoptic area. This is the region, an area which receives signals on warm sensors from the external and the internal world. And under this influence, it acts on the thermosensitive pathway just to regulate it. Well, when you have the action of neurokinin through its receptors on the MNPO, you have an increase in the dissipation of heat. But I told you, this effect is mitigated by the action of the estrogens. At the menopause, what happens? Well, estrogen disappear, most of them. And in that case, what you could see, and that has been seen at necropsies of postmenopausal women, is that this candy neuron nucleus is increasing to 3 times its size, more or less. And the result is that there is a lot of neurokinin B, which is secreted and which acts, of course, in heat dissipating activity, which is abnormally high, with vasodilation, perspiration. We are in the domain of hot flush, exactly in the domain of hot flush. So the idea was to use antagonist to this receptor, block any action of neurokinin B on this activity on the thermoregulation, and just to inhibit this dissipation of too much dissipation of heat. So we have the basis for understanding all that. And just to come to a non-hormonal treatment of hot flashes using NK1 accessory, but essentially neurokinin 3 receptor antagonist. And we got them because some 25 years ago, this kind of receptor of antagonist has been used in order to develop drugs against schizophrenia, but they failed, and they were left on the shelf. And they were taken back just to see if they could do the job of decreasing the hot flashes, which happened with pavinotant, which decreases VMS very rapidly and to an interesting amount of reduction, but increase in liver transaminase urged to stop these studies, and they were removed from development. So we turned to visolinetant, and the first work was done in Belgium, in fact, and then taken over by Astellas almost everywhere. And with adapted doses from, well, let's say 45 to 90 milligrams, you can have a decrease in VMS, which is rather important and statistically significant, and occurring very soon after the administration, which is also a daily one. But you have still there some treatment-emergent adverse events, such as frequent diarrhea, headache, liver transaminase, but under 3 ULN, upper limit of normal, not reaching criteria for liver injury according to holy FDA. The phase 3 is now terminated, but we have not the full results of this study. However, it seems to be a good one because Astellas is preparing for a submission to the FDA, but we have not the results exactly, and we want to be careful about treatment-emergent adverse events. And another one who is under study more recently, elinzanetant by Bayer, is also having a good effect on reduction of vasomotor syndrome in phase 2, but is now only with also some frequent diarrhea and headache, but not too much acting on the liver apparently, and we are only entering phase 3 at the time being. What does it mean? Is that the one who is more advanced, fezolinetant, is a rapid and effective, has a rapid and effective reduction of VMS and a related improvement in quality of life, including sleep, but also safety, and I put it in italic, seems potentially acceptable at least for 1 year. You know that if you are suppressing hot flushes, it may be for many, many years. So let's be careful, but let's say maybe it's all right. Indication, of course, the non-hormonal treatment of VMS in postmenopausal women with absolute contraindication to estrogen therapy. That means women with breast cancer or other hormone-sensitive cancers, increased thrombotic risk, we are back on that, side effects with HT, women not accepting hormones because many are still due to the negative action of the Women's Health Initiative study in 2002, that is exactly 20 years ago. And so they could fill this particular place. But when you compare to the estrogens, it's not the same story. According to the usual indications, estrogen therapy, including, of course, as we have demonstrated, Estetrol, is not only effective for treatment of VMS, but also for the full spectrum of menopausal symptoms and problems. Health-related quality of life, including quality of sleep and many other things, genitourinary symptoms and sexual problems relief, mood and cognition enhancement, improvement of some neurodegenerative diseases, bone preservation, decreased joint pain, skin, hair, body image enhancement, and this last point is very important to women also. And these are potential additional benefits not attainable with an NK3 receptor antagonist. I thank you for your attention.
Thank you very much for this extremely interesting lecture that we all learned from a lot. Thank you. Questions from the audience first. Yes.
Really, thank you for your presentation. In your phase 2 study, those findings study, you tested from 5 to 15 milligram, and afterwards in your phase 3, you choose 15 and 20 milligrams, which is quite unusual. You tested a higher dose as tested in phase 2. So what is the reason for this? I suspect that FDA is asking for 2 dosage as usual.
We did that in prevision of any demand from the FDA, because in the phase 2b, from 2.5 to 15 milligram, it was looking for the minimal effective dose. And this minimal effective dose, when you speak of hot flushes, is 15 milligram. And okay, we had a good idea that lower doses were not as correctly effective on that point. Turning to 20 milligram would show us all aspects of the statistical significance versus the placebo, but also the clinical significance as asked for usually by the FDA. So we had a larger margin of efficacy on the VMS in terms of frequency, in terms of reduction of severity as well, when going into that slightly increased dose. So we have a whole panel from a very low dose to a dose which is a minimal effective, and we have seen that the full spectrum is covered by that, but the 20 milligram is also bringing even more reduction, and it may be more interesting for people who are less sensitive individually to the action of Estetrol on their hot flashes, also with having that possibility of the 20 milligram. But at the same time, don't forget that we interrogate other endpoints, of course, with both types of doses of treatment. That was the reason why.
It is quite unusual to test the higher dose in phase 3 as compared to phase 2, for safety reasons only.
Yes, because we thought to fully answer the FDA demands on the clinical significance, even more than the statistical one. You know, it's like St. Thomas.
More questions from the audience? Or do we go to the other space?
the fifth phase.
The question? So other states maybe are driving to Knock, I don't know, somewhere like this, because they are not answering questions. But I have a question. May I ask it? Like a woman question, you know, like fascinating debate for me. Aging looks so difficult. Though Professor Genazzani said women will not age, we have to talk about changing and not aging. I heard a lot about what is getting inside, not what is appearing outside. So I just saw some point saying that there was a skin improvement. What will be the difference from if someone is aging with Estetrol instead of another pill? What will be the difference? Will women have a better quality of skin? Maybe age less quickly? Do you have data already?
What happens inside when you are taking Estetrol, for instance, at the time of menopause? Well, you have a very interesting action on your metabolism. And your metabolism is aging with time. And that means that it maintains a normal level of the lipids, an improvement in your glucose metabolism. And you know that long studies have shown that taking estrogens diminishes the initial development of diabetes type 2 in around 30% of women. Other action is the atheroprotective. Whoa. Protection against atherogenesis. And it has been shown in many ways in any type of animal, but also in the human being, that you decrease the risk, the cardiovascular risk with using estrogens. And particularly, you see, if you are using an estrogen that does not increase coagulation. Okay, that is very important. And it may prolong life. And it has been shown that the longer time you take an estrogen at the menopause, the more life is prolonged. So we are going on the skin, on the image of the woman. Terrible change. When you look
that's essential
at women waiting in your waiting room, there are 5 or 6, and you see there are 2 who are taking estrogens, and you know exactly, not remembering what you are giving them, but sure that because their appearance is much younger than they should be. Well, this is an image. Brain function, you have here what we said about connectivity, about psychodynamism and so on. It's also the case. Well, can we go further? No, I think this is already a very good demonstration.
Okay.
I would want to complete that by saying that
we will give you a microphone, Professor Foidart, so that everyone can hear you.
Jean-Francois Arnault, who is sitting close to me, has done studies on the skin and on the wound healing with estradiol, not with estetrol, where he shows that the estrogens increase the wound healing and the scarring, and it's mainly through improved angiogenesis in the wounded skin. Remember that you can do fetal surgery into the uterus, and intrauterine fetal surgery leads, when the baby is born, there is no scar. There is a perfect healing of the scar tissue. When an adult, you have always ugly scars. During intrauterine life, you have no scarring process. We hypothesize that E4 could have a beneficial impact on the wound. We have ongoing studies supported by Mitra in Florida with pigs, where we evaluate the impact on the wound and on the skin. And Graham Dixon, who is the CSO behind me with Maud Just, have designed a study that will be performed at La Charité. La Charité is the big academic hospital in Berlin, and they are going to evaluate the impact on the skin, on the wrinkles, on the skin, on the thickness of the hair. And we anticipate to have a very beneficial impact on the skin also, but that is for the future, and that's going to be evaluated in clinical settings as well.
Could be also a project of cream, something that you could put as a cream on the skin? For the wound? No, but I mean, I'm sorry, I'm getting deeper.
If you want to have it for compassionate personal use, we can give you some.
That's not the purpose, but if other women are interested, I can make a list.
The thing we forgot was the bone. Osteoporosis is very important, up to fracture, as you know. Estrogens, and is the case also for Estetrol in terms of its action on reducing bone resorption, is having a preventive action and even a therapeutic action on osteoporosis.
Thank you, Professor. And if you want to send your names and email addresses for the Professor Foidart cream, you can send them to me. Don't worry, I will transfer. Mr. President.
Yes, I was thinking that perhaps you could announce the points of INAMI.
Oh, yes. You're right. Thank you so much for your help. This session has been accredited by INAMI as usual. So if you want to send your emails, you can send them as of now to the address that you know already and that you will see appear on your screen. contact@armb.be in French, contact@armb.be. Of course, if you are not member of the Academy and if you are a doctor watching us on YouTube, you can send your data, so first name and number of INAMI number, sorry, so that you can get everything you need. Thank you so much for being with us today, and I'll give the word to the President.
Thank you very much. This was a wonderful session. It will be memorable. We all learned a lot. And I would like to end with a couple of things. First is to thank Jean Michel Foidart for putting together this wonderful scientific program. To say that if other people on the Zoom or the YouTube desire to ask questions, they should address them to the Academy. To the website of the Academy, there is an email there. And I'm sure that the speakers will receive those emails and will diligently answer those questions, because this is a subject of great medical interest. I would like to say that the next session of the Academy will be next week, next Saturday, the 30th of April. That will start with an éloge académique of Professor Antoine Bremer by Jean-Claude Pector, and then we will have a session which we called diverse session. That is, there are contributions from different areas of science, from Melody Schwartz from University of Chicago on on lymphatic network, from Frédéric Kridelka on cervical uterine cancer and lymphangiogenosis, Alain Chariot, molecular oncology, and Laurence Ries, limits and potentialities of retinal implants. And then I would like to say, last but not least, that on May 7, we would have a dedicated sort of symposium on breast cancer with 2 parts. This is organized by Professor Frédéric Le Couvet from Brussels and Martine Picard from Institut Bordet. We will have speakers such as Monica Morrow from Memorial Sloan Kettering, Nicolas Lemaire from Institut Gustave Roussy, David Pasquier from Lille, Martine Picard from Bordet, François Duhoux from Saint-Luc, Frédéric Lecouvet also from Saint-Luc, and Philippe Aftimos from Institut Bordet. So that is on May 7. And last but not least, we invite you now for a cocktail in honor of Jean Michel Foidart and the wonderful speakers of today. Thank you very much.
Compliments for the beautiful